Hello everyone, my name is Aishwarya Gosavi and my roll number is 7509. As all of you have read the title of this video, which suggests causes of DNA mutation by chemical mutagens. Yes, let's understand chemistry behind this biological phenomenon. So for this, first we have to know what is mutation. So mutation is change in the sequence of DNA. Mutagenesis. It is a process by which genetic information of an organism is changed and results into mutation. There are two types of mutation, spontaneous mutation and induced mutation. Spontaneous mutation is also known as naturally occurring mutation. Example of such mutation is cancer and various hereditary disorders. And whereas induced mutation. It occurs when an organism is exposed either accidentally or deliberately to mutagenic agents that interacts with DNA to cause DNA mutation. Induced mutations typically occur at much higher frequency than the spontaneous mutation. There are three mutagenic agents which can cause induced mutation. First is physical mutagens. UV radiation and other high frequency radiation are examples of such physical mutagens. Biological mutagens. Viruses and bacteria can be called as biological mutagens. Example is HIV. HIV is a causative agent of AIDS, and this HIV virus can incorporate its own DNA into our DNA and causes our DNA to get mutated. And chemical mutagens. Chemical mutagens. Chemical mutagens are naturally occurring or synthetic chemical that can cause mutation by changing bases in DNA and which can also insert base pairs or change base pairs and ultimately causing frame shifts mutation. Before understanding the mechanism of how chemical mutagens can cause DNA to damage, let's brush up the basics. As we all know, DNA contains deoxyribose sugar, phosphate groups, and nitrogenous bases. There are two types of nitrogenous bases, purines and pyrimidines. In purines, we have adenine and guanine, and in pyrimidines, we have cytosine and thymine. Adenine pairs with thymine by forming two hydrogen bonds, and guanine pairs with cytosine by three hydrogen bonds. And all these nitrogen bonds ultimately forms double helix structure of the DNA. This was all about the basics. Now understand the different types of chemical mutagens. So there are four types of chemical mutagens. First is base analog, second is base modifying agent, third is alkylating agent, and fourth is intercalating agents. So first, base analog. Base analogs are nitrogenous bases which are very similar to the normal nucleotide that synthesizes the DNA. Like normal bases, base analog exist in two tautomeric form, normal form and rare form. And in each of the two states, the base analog pairs with two different normal bases. Then they may get incorporated the DNA in place of normal base. So example of base analog is 5-bromouracil. 5-bromouracil is a base analog of thymine. Let's understand by looking at the structure. So this is the structure of thymine, and on the fifth residue of thymine we have methyl group. But on the base analog of thymine, which is 5-bromouracil, we have bromine group on the fifth carbon. So this minute change in the functional group can lead to formation of base analog. But how does 5-bromouracil induces mutation? So it induces mutation by switching between its two chemical states once the base analog has been incorporated. To understand this, first we have to see how does it pair with other nitrogenous bases. So in normal state, 5-bromouracil resembles to thymine and thus it pairs with adenine by forming two hydrogen bonds. But in rare state, it resembles to cytosine and thus pairs with guanine by forming three hydrogen bonds. So this was the pairing. Now let's understand the mechanism by which it damages the DNA. So during the replication of DNA, enzyme DNA polymerase adds new nucleotides in the DNA strand. But in case of mutation, DNA polymerase accidentally add 5-bromouracil into the growing chain of DNA. But problem is not here. The problem will arise when this DNA strand having 5-bromouracil in it will be used as template strand. So in the next round, when the replication where the template strand used contains 5-bromouracil, then this 5-bromouracil will shift its shape from resembling to thymine to resembling to cytosine. And thus, it pairs with guanine in this second replication phase. And this causes TA base pair to change into CG base pair. This is a transition mutation. So 5-bromouracil can also induce CG base pair to change into TA base pair and causing transition mutation that way. If it gets incorporated in the first round at cytosine and pairs with guanine in them. So this is how two types of transition mutation can occur by single base. So let's understand base analog thing in a good note, that is AZT. AZT is also an example of base analog. AZT means azidothiamidine. It is a base analog of thymidine. It can also use as drugs in the patient of AIDS because AZT do not have any proper linker, hence it stops the DNA to get replicated, and it can be used as drug in the patient to stop the replication of HIV virus. So this was all about base analogs, and let's understand second category of chemical mutagens, which is base modifying agents. As the name suggests, base modifying agents are the chemical agents which can modify the nitrogenous bases. The formal definition is the chemicals which can act as mutagens by modifying the structure and properties of bases. There are two types of base modifying agents. First is nitrous acid and second is hydroxylamide. Nitrous acid. Nitrous acid acts as a deaminating agent. Now, what is a deaminating agent? Deaminating agent is an chemical agent which can deaminate a chemical structure. That means it can remove the amino group from that chemical structure. So the deaminating agent removes amino group from bases like guanine, adenine, and cytosine. Let's understand its reaction with guanine. So when nitrous acid reacts with guanine, it removes the amino group from this carbon and adds an oxygen group. This makes up the xanthine as a product. The formed xanthine now pairs with cytosine only. So same pairing occurs as guanine. Hence no mutation occurs in this case. But when nitrous acid reacts with cytosine, it forms uracil as the product. And this uracil will now pair with adenine instead of guanine, and hence this results into CG base pair, TA base pair. This is also a transition mutation. And when nitrous acid reacts with adenine, it deaminates the adenine by removing amino group and adds up oxygen group and forms hypoxanthine. So this hypoxanthine, which is now formed as a product, will pair with cytosine instead of thymine. Hence, this reaction of adenine with nitrous acid results into AT base pair to change into ZC base pair. This is a transition mutation. Now let's understand the second base modifying agent, which is hydroxylamine. Hydroxylamine is a hydroxylating mutagen which is specifically reacts with cytosine. Now let's first understand what is a hydroxylating agent. Hydroxylating agent, which is a chemically compound which adds hydroxyl group to a compound and modifies it. So it modifies cytosine to hydroxyl aminocytosine by adding hydroxyl group to cytosine. So this hydroxylaminocytosine now formed as a product will pair with adenine instead of guanine, and hence the mutation uh, uh, is formed as CG base pair to change into TA base pair. This is a transition mutation. This mutation cannot be reverted back by second treatment with this chemical only, but it can be reverted back by using chemical mutagens such as fibromyuris, which causes TA base pair to change into CG base pair, and hence the mutation effects can be nullified. Third category of chemical mutagens are alkylating agents. So alkylating agents means chemical compounds which can alkylate. That means which can add alkyl group to the chemical structures. So the methyl methane sulfonate MMS is a group of alkylating agents that introduce alkyl group into bases at number of locations. Examples of alkyl groups that can be added by MMS are methyl group and ethyl group. Most mutation caused by alkylating agents results from addition of alkyl group to the 6 oxygen of guanine to produce 6O6 alkyl guanine. After treatment with MMS, some guanine are methylated to produce O6 methyl guanine. It can be seen here. So this is the structure of guanine, and on treated with MMS, we have now O6 methyl guanine. And this O6 methyl guanine will pair with thymine rather than pairing with cytosine. This gives GC base pair to change into AC base pair. This is also a transition mutation. So the fourth category of chemical mutagens is intercalating agent. Recall when our professors instruct us or warn us about proper handling of ATBR during the experiments of agar electrophoresis. So, what does ethidium bromide do to our DNA? How does it damage us? Let's understand that. The intercalating agent inserts themselves between the adjacent bases of the DNA and it causes the DNA double helix structure to get relaxed. Examples of such intercalating agents are acridine, proflavin, and well-known ethidium bromide. There are two ways that can be used by intercalating agents to damage our DNA. First way, if the strand which does not have intercalating agents between its base pair and is used as template strand during the replication process, then an extra base is inserted into newly synthesized DNA strand opposite to the intercalating agent. Because the intercalating agent form an empty space in the strand and hence during the replication process the DNA polymerase will randomly choose any nitrogenous bases and add it here. But during the second round, this strand, which does not have intercalating agent, will now add subsequent nitrogenous bases, and the overall effect is per addition mutation, and this can ultimately leads to frame shift mutation. The second way by which intercalating agent can cause mutation is if the strand which have intercalating agent between its base pair and is used as template strand for replication and strand replicates after the intercalating agents. Cause this result into base pair deleting mutation. This ultimately lead to frame shift mutation. Uh, so the as intercalating agents can either cause addition of base pairs or deletion of base pairs. Its treatment with the same intercalating agent can revert the intercalation effects. So, but this does not mean that we can use ATBR carelessly. We have to be careful. So, thank you for listening to me. And these are the references which I used to make this PPT. Thank you for watching patiently. Now, like the video.